Hi everybody, this lecture is about patterns of inheritance or Mendelian genetics. Now some of you might be wondering why we would need to go into such detail on Mendelian genetics, and this is actually one of the most important components of understanding one of the four pillars of modern biology, which is evolutionary biology. To understand evolution, you need to understand population genetics. To understand population genetics, you need to understand the genetics of individuals. Besides which, as humans, it's quite interesting to know a little bit about Mendelian genetics. If you're trying to figure out whether or not your kid's going to have dark hair and dark eyes, or light hair and light eyes, that's always quite fun. But then there are also some features that are of great medical importance. So if someone in your family has got a genetic disorder, you might want to figure out whether or not you have it, or you can pass it on to your children. So variation is something that's found in many populations, and when we describe a wild type trait, that's a trait that's most commonly found in nature. So traits are usually inherited in particular patterns. So if you took the yellow budgie from this picture and you mated it with other yellow budgies, chances are you'd have a higher likelihood of having a yellow budgie baby. So it turns out that yellow is the wild type. That's what you'd find if you go out into the bush in Australia. You'd find wild yellow budgies. So if you took two parents that were both wild type, your expectation is that all of your offspring would be wild type. On the other hand, if you took a wild type budgie that was yellow and you mated it with a sky blue, a very rare sky blue, what you'd find is that first generation offspring were all wild type. But if you bred siblings from that first generation offspring together, what you would find is that some sky blue individuals would reappear and the wild type individuals would be dominant in a 3-to-1 ratio. That tells you something about the sky blue trait versus the yellow trait. Now, one of the first people to come up with a systematic approach to studying inheritance was Gregor Mendel. He was actually a contemporary of Darwin, but they never got to meet each other. His work was primarily done with pea plants. Peas are particularly useful for these sorts of studies because they can self-pollinate. What he would do is he would take a flower right before it was ready to pollinate itself, and he would actually castrate some of the flowers. So he'd remove the male parts from some flowers, the stamens, and then he would deliberately take pollen from other flowers and pollinate the female parts. So he could choose which parent for which sort of breeding cross. He would allow the eggs to develop. They develop into peas, and each pea in a pod, you could think of it as a sibling, so it's like a litter of peas, and then he would plant those peas and have a look at their traits. So he created what we call true breeding varieties of plants. These are plants that if you have a purple flower and you mate it with another purple flower, you will only ever get purple flowers. In modern biological terms, we would call this homozygous for the purple trait. So then he did a series of tests where he would breed a true breeding purple with a true breeding white and have a look at the results. So he tracked several characteristics, including color, position of flower, seed color, seed shape, pod color, and stem length. And what he found was whenever he bred a true breeding for one character with a true breeding for another character, one of those characters dominated the others, and in the F1 generation, all of the offspring resembled the dominant trait. So if you took a flower that was an axial flower and you mated it with a flower that had a terminal flower, all of their offspring would be axial. So we would call that a monohybrid cross. So that's a cross between parents that differ only in one characteristic. So that allows you to follow that one characteristic. And Mendel's monohybrid crosses would always result in the F1 generation having all the plants resembling the dominant characteristic, and then when you crossed F1s with other F1s, you would end up with a 3-to-1 ratio of the dominant character to the recessive character. He developed some hypotheses from this monohybrid cross. The first is that there are alternative forms. We call these alleles. So genes, which are sequences of DNA that make proteins, these genes can come in different forms. For each characteristic, an organism has got 2 alleles. So that means that I can have a maximum of 2 different versions of a gene. It's because I'm diploid, I carry 2 full copies of my genome. Now, you can actually have more than 2 types of alleles. In some cases, you've got 3 alleles, like in blood typing. You can have 20 or 30 or 12 or 5 different alleles, but at the most, each individual can only carry 2 alleles. Gametes, which are sperm and egg cells, can only carry 1 allele each. An allele can act in a dominant or a recessive manner. Now, that was Mendel's finding. Alleles can actually behave in slightly different ways. So, Mendel's statement that alleles can be dominant or recessive is what we would call simple Mendelian inheritance. There are cases where alleles neither dominate each other, and that's called incomplete dominance. And then we can also have a case where alleles are both dominant, and we call that codominance. So an explanation of Mendel's results can be described using a Punnett square. So if you look at a pure breeding parent, such as a big P, that's a purple parent, and there's an error in this diagram, a little p parent, which would be a white parent. So the gametes that the purple parent could make would be a big P gamete. The gametes that the little p parent or the white parent could make would be little p. The F1 generation, all of their genotypes would be big P, little p. That means that the F1 generation can make 2 types of gametes, either a big P gamete or a little p gamete. And to look at the possible offspring that those F1s could make, you could draw out a Punnett square. It's also known as a contingency diagram. It illustrates the possible combinations of offspring. Oof. So if you could have a big P sperm mate with a big P egg, you would have an individual that would be big P, and it would end up being purple. If you had a little p egg with a big P sperm, you would end up with a heterozygote, and so you would have a purple individual. Once again, a little p sperm with a big P egg gives you that heterozygote because it's got a big P, it's got a dominant allele, and it's going to be purple. Or you could have a little p allele and end up with a white one. So we can describe 2 ratios, a phenotypic ratio. This is the ratio of the phenotypes. The phenotype is what does it look like. So in this case, is it purple or is it white? And there are 3 purple and 1 white. The genotype ratio is the ratio of the different types of genotypes. And we normally do it in order of big P, which we refer to as homozygous dominant. Homo means same as, zygous means to make the egg. So we can make 2 eggs that are the same as each other, but they've both got dominant alleles. Thus, it's called homozygous dominant. You can have homozygous recessive, little p, and then you can have heterozygous. Hetero means other than. So we always do it in the order of big P, little p, little p. And in this case, the ratio is 1 to 2 to 1. And that's a fairly stereotypical result that you would expect if you have 2 heterozygous crossing with each other. So Mendel's principle of segregation is that the pairs of alleles separate during the formation of gametes, and the fusion of gametes at fertilization creates the allele pairs again. So homozygous means an organism has got identical alleles for a gene, so you could have homozygous dominant or homozygous recessive, and heterozygous is when an organism has different alleles for a gene. A test cross is a special case where you have an individual who's got the dominant phenotype. So you have a purple flower, but you don't know, is it homozygous dominant or is it heterozygous? What you would do in this case is you would mate your unknown purple flower with a white flower. Because what would happen is if the purple flower was homozygous dominant, or big P, when you mate that with a little p flower, you're going to end up with all of your offspring being purple. On the other hand, if your purple flower was a heterozygote, big P, little p, what will happen is half of your offspring will be purple and half of your offspring will be white. So you can look to the children to indicate as to whether or not the unknown parent was homozygous or heterozygous. Now, in probability, we have slightly different rules of multiplication than in the sort of regular life. So you would be looking at what is the chance of having a big B and what's the chance of having a big B from the other parent. So when we say the chance of having this and the chance of having that in probability, we actually use multiplication instead of addition. And so that's why the chance of being a big B is actually 1/4, because you have a half of a chance of getting a big B from parent 1, half of a chance of having a big P from parent 2. A half times a half equals 1/4. So Mendel had a second principle, the principle of independent assortment. And this is simply that, sorry, I'm stepping ahead, that if you look at two traits simultaneously, such as both color and shape, you can make the assumption that the two genes are actually on separate chromosomes and they behave independently of one another. This is generally true. There are going to be cases in more advanced genetics where you would have to reject the assumption of independent assortment. But for your level of biology, if you're looking at more than one trait, you can actually consider them separately from one another. Now, this example here is called the dihybrid cross. This is looking at parents who have two traits, and they're homozygous for each trait. So you've got a parent who is dominant for both color and shape. So they're round and yellow. Their other parent is recessive for both color and shape. So we have a wrinkled parent who is green. Now the first parent can only make one type of gamete, a big R, big Y gamete. Now how do we determine what the gametes are? Well, remember, you're going to need at least one gene for shape and at least one gene for color. So when you look at that parent, you could say you could have one big R with this big Y, this big R with that big Y, this big R with that big Y, that big R with that big Y, but it ends up being the same. You only have big R, big Y as your parent. You could also have little r and little y. That's your only gamete that you could make from that parent who is a homozygous recessive for both traits. Now, when you combine those gametes, you end up with an individual who's heterozygous at both traits. So they have a big R, little r. Because they've got a big R, they're round. Big Y, little y, they're heterozygous, but because they've got the big Y, they're yellow. Now, if you took that individual who's heterozygous at both traits and crossed it with another individual who is heterozygous at both traits, what you would end up with for possible offspring is this crazy big Punnett square in front of you, where you end up with 9 out of 16 of your offspring are yellow and round. So that is that they're dominant for both shape and color. 3 out of 16 are dominant for shape and recessive for color. 3 out of 16 are recessive for shape and dominant for color, and 1 out of 16 is recessive for both shape and color. And this 9:3 to 3:1 ratio is a stereotypical result that you would get if you have two parents that are heterozygous at both traits. We also use family pedigrees. These show the history of a trait in the family, and they allow researchers to analyze human traits. So typically we use a circle to represent a female, a square to represent a male, and we color in the shape if the person has the trait that we're interested in. And this allows us to go through the family tree and establish who's heterozygous and who is homozygous. Now, a lot of figuring out a family pedigree has got to do with determining whether or not you are lucky to have a test cross. So in this diagram below, it turns out that being deaf is homozygous recessive. So anytime you have a homozygous recessive person, or a little d person, and they are having offspring with an individual who has hearing, that actually counts as a test cross. So Jonathan and Elizabeth, if Elizabeth was big D, none of their children would be expected to be deaf. But because two of their children, and honestly, you only needed one child to be deaf in order to prove the point that Elizabeth was actually heterozygous, because she would have had to be giving a little d to her child in order for the child to be deaf, because husband Jonathan can give a big D sperm, she can give a big D sperm or a little d sperm, and if she gives little d and he gives little d, then you have an individual who's also deaf. There are many human disorders controlled by a single gene. Some of them are recessive disorders like albinism and cystic fibrosis, galactosemia, phenylketonuria, sickle cell disease, and Tay-Sachs disease. In this case, if you have a little a, you've got the disorder. The heterozygous case and the homozygous dominant case is someone who does not have the disorder. Dominant disorders include achondroplasia, Huntington's disease, and hypercholesterolemia. And in these disorders, a heterozygous or a homozygous dominant individual will have the disorder. If you, for example, if you are of normal height and you do not have dwarfism as a result of achondroplasia, then what that means is that you are homozygous recessive. Most human genetic disorders are recessive. So that means that the disordered individual is homozygous recessive. Heterozygotes are what would be called carriers, so they are normal in their phenotype, but they are actually carrying the disorder. There are exceptions to Mendel's rules. One of them is incomplete dominance, which you can find in plants and in people, and that's when you actually have the third phenotype. So in simple inheritance, you've got only two phenotypes. You've got red and white. In incomplete dominance, the heterozygote has a phenotype that is in between both the dominant and the recessive. So here you have a pink phenotype. It's quite common in plants. Another non-Mendelian inheritance pattern is the ABO blood grouping. This is where you have multiple alleles. There are 3 possible alleles, and they can interact with each other in a codominant manner. If you are blood group O, that means you've got 2 copies of the little I allele. If you are blood type A, you're either a homozygous A or you're a heterozygous A. So that means that you've got 1 little I allele and then you've got 1 A allele. But the A allele is dominant to little I allele. And likewise for B. However, if you are a super lucky person and you've got an AB blood grouping, that means that you've got both the A and the B genotype, and you're expressing both of those simultaneously. Pleiotropy is when one gene affects more than one characteristic. We can find this in examples like sickle cell disease, where you have just one allele that has an incorrect ordering of nucleic acids. It results in a single protein that is non-functional or only partially functional, and it can result in a large number of possible issues ranging from kidney failure to anemia, pain and fever, spleen damage, etc. This is actually quite common, but pleiotropy is when one gene affects more than one characteristic. Polygenic inheritance is when you have a trait that appears to be continuous. So it's not like you just have two options, tall and short, but you've got all sorts of grades of in between. This is also found in skin color. And it's where you have two or more genes that all work together on a single phenotype. So in this example here, you've got a very light parent with a very dark parent, and as you might expect, that first generation has got moderately toned individuals. But if you have two moderately toned individuals who have children, there is a chance that some of their kids would be super, super pale, and there is a chance that their kids would be really, really dark. So even within the same family, if you have two moderately toned parents, you can have some children coming out very light and some coming out really dark. In humans, we determine our genetic sex with sex chromosomes. They're designated as X and Y. Females have got two copies of the X chromosomes, and males have got a copy of X and a copy of Y. So it's actually men that determine the sex of their offspring. There are some disorders that result from sex-linked genes. That means that those are genes that are carried only on the X chromosome. Red-green colorblindness is a really good example. So on the screen you should see a number 7. If you don't, you might want to go online and test yourself for some colorblindness. What happens is on your X chromosome you've got a gene that codes for a protein that is used in light detection, and if you're a male you only have one copy of that gene. If you're a female, you've got two copies. Oops. If you are a male and your one copy is broken, you'll have red-green colorblindness. Females, on the other hand, if one copy of the gene is broken, they have another copy on their other X chromosome, and so they'll still be able to make the protein that's needed to be able to see. So red-green colorblindness is actually much more common in men than in women, because a female would have to inherit two copies of the defunct gene in order to not be able to make the protein required to be able to see color properly. So I hope that this helps you a little bit with Mendelian genetics. This is a topic that requires quite a lot of practice. The SymBio labs should help to give you some of that practice, and if you read through your notebook a few times you'll get an idea of how people work problems in Mendelian